This is going to be on the prokaryotic replication fork. We'll start with our origin of replication, which is here and also here. The origin of replication is going to be a specific nucleotide sequence that allows a certain protein to bind to it to start the replication process. We'll start with gyrase, which is going to be located here, and this basically helps to unwind and relieve any stress and coiling that may occur. We then have our helicase, and this actually comes in and it opens our fork. And it'll open two forks because our DNA is going to open in a bidirectional manner. The helicase does this by breaking the hydrogen bonds between the nitrogenous pairs. After that, we will have the single strand binding proteins, and they will come in and attach and basically keep the strand from recoiling back into the helix structure. After that, we'll allow the DNA polymerase 3 to come in. And a few things to know about the Pol 3 before we go on is polymerase 3 can only build in the 5' to 3' direction. It cannot go in the 3' to 5'. Also, it has to have a 3' OH group to attach to, or else it's just not going to be able to. So for this strand here, and also down here, which is going to be known as our leading strand because it's continuous, it's able to attach to the 3' OH group and the 5' side, and it's actually going to go down and lay down all the nucleotides one by one. Now, on the other side, because DNA is anti-parallel, it's going to be running 3' to 5'. So to kind of combat that and help the DNA polymerase 3, we have something called primase. That's going to go in and it's going to add RNA primers, which is going to allow the DNA pol to attach to it and lay down those nucleotides. After that happens, DNA polymerase 1 is able to come in, replace those RNA primers with DNA. Now, because of this, how it's set up, it basically has to wait until the fork opens, so it ends up being in fragments. This is called the Okazaki fragments. And to complete this, basically we have the enzyme ligase that will come in and it'll attach or connect all of the fragments. And it does this, actually everything works, it needs energy, and all the energy comes from the breaking of the phosphate bonds on the nucleotides, and then it creates new phosphodiesters bonds with the new nucleotides and the existing strands.